Enzyme. So, go my name and turn knock and wale at the WE the Success Lecture. Actually, I may welcome you. Welcome to the WE the Success Lecture. So, channel and content. Saloon jokes. Min turn knock and wale writing enzyme. So, when we say enzyme take a duck to one to ban duck to ban what again. But what is the most important initially is that I need you to subscribe as I do before. But you know that is a long need one. Because you know that long young gun Martin wanting to channel share support. Now go comment kuntan le tenke channel at WE the Success Lecture. Now go reply ni kunbo tin la top yen kabo whenever just reply to you. You can also reply to me. So they makes me happy. So introduction. Nama enzyme chakan ka bogan new and bogan jidit ka grade 11 actually. I mean I think it's grade 11 actually. So guame we have enzyme. So generally, you know, you may have heard the word enzyme. So me ko non ko na actually it may be difficult. Tan and ko nan ka ko no da ko ndaru enzyme da ran me na ko no directly. But just enzyme ka kan tin di alter konta ko metabolism ne dio chi video metabolism don chi gut. Me che jen ko video metabolism ka catabolism ka anabolism. Like all those processes, the alter the poiran including kon chama ba digest babara. So quite digest enzyme tin ta digest enzyme ta kalon ta tin ta pancreatic enzyme ta tin ta enzyme ti ta ke brood border la ka jen intestinal cell ta tin ta enzyme to ko release ba ka stomach ba enzyme ti dele jabo ne ka salber gland ka salber amylase. So can different enzyme they have different function, and their function is that they just only speed up the reaction. But when an enzyme may be defined a biocatalyst, it's assigned by the living cell. That means when we say biocatalyst, it means that that can integrate a living organism. Say, for example, you are a living organism. You have a certain enzyme in your body to take a letting one who can carry out for you. You can go out just take it to one. She and Kokon Bumichi number, take a ball or nang, don't want glucose out of Mitchell, the long daily toba, liver, no molecular glycogenesis. Glycogenesis take an enzyme, particularly to like enzyme limiting then which are glycogen synthase. So it only a different enzyme. So enzyme a biocatalyst, exactly. She comes out of fine government. So a biocatalyst milk because it's a living organism. So the most important that I need you to take into consideration when go brana capable enzyme they are protein in nature. Enzyme they are protein in nature. When look on woman our most enzyme, but not all enzyme. Remember, chai jota enzyme they are protein in nature, but it's not all enzyme. So I know most enzyme in their nature time chocolate get protein. They are contact different nutrient. We have carbohydrate, we have protein, we have lipid, water, minerals, vitamins, calcium, protein. So different protein ten ko consume kan tam protein te lat ke liver ke. So me tare de no lat yo kan ba ti yo ngutin latin akan ba enzyme ne point ba ko latin to ten bitede ten be different metabolism point be kan carry out. So except there are some exception. All enzymes they are protein in nature, but there are some exceptions. For example, RNA enzyme, which are ribosome. To DNA, but take a enzyme. And those enzymes, they are what? They are not protein. They are nucleic acid. Nucleic acid, you can't go a fat nutrient, can like macronutrient. So except can got different enzyme. All enzymes they are protein except this enzyme. Except that RNA take ribosome. Ribosome enzyme like RNA polymerase, transcription, protein synthesis, so they are colloidals actually. So when we say they are colloidal, then mean they are solution to grade 12. When we say colloidal solution, it's kind of solution settle down. A solution may be a way like it is not exactly very saturated. But there go. Good example, check a barret. Which you go call a bar, which I'll call which you want to pal in a go. I get a well read. So that means local new consider. So enzyme they are colloidal. One they are protein nature, they are colloidal, and they are thermolabile, very important. When you say they are thermolabile, that means they can be break down by heat. If you are in serious high temperature, let's say, la me enzyme which let go at a very high temperature la belel belu chebe let go chebe function la be nature la so ko protein la ko nature go at a high temperature raude diolben and is specific interaction. Don't forget this. Each enzyme is specific color and balat. For example, let's say which our pancreatic amylase which were a very specific. When a carbohydrate do which a breakdown to do, but don't bend by the stomach where at so by breakdown will mean to amylofectin can amylase. Don't be carbohydrate be dollar with. So go mane amylase can amylofectin ka carbohydrate that immediately time with you down here. So which you with pancreatic amylase lad be one other well digest need amylase corrode. Chadam and what bong digest Jamichuale amylofectin chapa digest. But digest mutual amylose. So a part no more. Then colaro enzyme is being character. You then ban a carbon enzyme are protein nature that are colloidal, some labile and specific interaction. An enzyme is globular protein, very important. So chikone enzyme different protein carton we have for example based on the time chakalat kije. Take a globular protein, fibrous protein. So enzyme take a group which are globular protein with uniquely shaped active site, and it acts as biological catalyst for a specific reaction, but remain unaltered after reaction. To go about information the other. But maintain your enzyme globular protein, take a own active site material like a react substrate and bakula digest substrate and besansign. Worker over gen catabolism can volume metabolism, either a catabolism or anabolism. There is no others unless if the enzyme that are out order command either both catabolism and anabolism, bakula chalumuno, chala anabolism. So what is important, even if the enzyme has carried out the function, which is function than a go to mission, a war better remain unaltered later on. Tell a bagario, Belane Laranamo betalk. Most enzymes are proteins, but RNA processing catalytic activity, ribozyme. Ribozyme enzyme peptide transferase, ribonuclease, RNA polymerase, and so this enzyme RNA, ribonuclease, peptidal transferase. DNA betega dwarbo michel deoxyribozyme, ligase. So biocatalyte can be protein, DNA or RNA, but most of the enzymes they are protein in nature. But and you remember the enzyme can be protein, can be DNA or RNA. But they tell meridian outside these three. The structure of enzyme. So enzyme has its own structure to go when teacher like but banako good can go wale bane kapad one and go bane tamdan bane nectin. So kakan structure of enzyme we have to learn them one by one. Each specific structure on lorma. Tell me you could good and door kelo. Then enzyme can quack ideal. So third, the structure yellow which a substrate molecule. Imagine substance bakula digest bakula catabolite ke banabolite as enzyme chali substrate. So guame agent area banja area machal active center agent center and go be substrate bakula banja. So guame neme mabar me can protein change it can acquire enzyme like a coenzyme protein. So they have their own function also. So this one chala catalytic group la neme substrate active site, but now take a catalytic site. Catalytic site make a catalytic group, agent bungola, agent function color, Namakubanian, active site could bind in the substrate, be me catalytic site, catalytic group, or color digest down. One away one by one. Active site, network active site, active site in the active center of a catalytic site and substrate binding site. So that means active center, you can active center agent area agent area in kulurum ke substance min kurum ke substrate dan bakule by enzymatic function bakule ka out kede. So take a guadro, take a catalytic site, take substrate binding site. Catalytic site catalytic side when you can and the funk got out the active site catalytic site and substrate binding site. Substrate binding site man be substance and bakule use by binding elaborate bare dop ka enzyme kede. Okay, So when catalytic side the region of enzyme that catalyze the chemical reaction, which is called then quality of the catalytic side, then go back chemical reaction. That is agent side which manipulate the substrate to help reaching the reaction transition state and the equilibrium faster. So Mugorko by reaction transition state or the equilibrium formation and Gorko Benautin. So whenever joy catalytic site then agent may be so it may be slightly separated from its substrate binding site, or they may be integrated in one another. Let's go back to this picture. So look sometimes then catalytic site let it substrate binding site. So sometimes they may be together or sometimes they may, but most commonly they are separated. The substrate binding site, catalytic site, the substrate binding site is the site at which substrate is specifically bind. Agent side and B substrate binding and activate the chemical reaction along with the catalytic side. Actually, the active side is not rigid in structure and shape. When we say active side, coactive side may rigid in structure and shape. Allow take a change to work your enzyme. So it is flexible to promote the specific substrate binding. Anyway, consider like Michi Balloon Michi Kuzika Michi let the Tedutin. She tell me like in so brother guy get a but a go bishop gentle take enzyme. Enzyme is flexible. Like she active side, she very straight. I mean, and gen active center in general. It's not very rigid. So it must allow an enzyme to have conformational binding. That means a lot of times you get the enzyme that are binding. They're going to take a don't see we just going to do biology well care well child take a mutually x take a mutual x model mutual ala ek can do them a lot getting a lot wrong turn on child la michi la ka muluro ka shape laboratory calabration don't get a to me tell na ko la ka ngon tong ta ko ta ke tin ko ko lot ko den ga la ki do ten jang ja yogo ne ta dangan fan. So that means janta ta enzyme ala jen be la very flexible ko bro lot ke dun dana. So the coenzyme or cofactor 3 ban go jo go 3 katila let kel ka enzyme o enzyme in general let the protein but ta katila go ri ja coenzyme cooperative ni ki da ko ni chal ko dol bilet ke ke ko mi chawa ku ramete yen kel to enzyme coenzyme do that in different mineral different vitamin content. So coenzyme or cofactor on which some enzyme depend are present as partly as catalytic side. Then mean coenzyme remember manubano nyame be ngai part of catalytic side. Substrate that bind active side by weak non-substrate bind at the active side by weak non-covalent bond. Yes. So consider like if the substrate bind with active side by covalent bonding, covalent bonding, a very strict bonding. That means that word that bind it is very difficult to remove it. So if it is very difficult to remove it, we don't have to form product. Common gorgo coenzyme, a web substrate by now modify by product by now. So there must be a non-covalent binding. That means very weak binding. Otherwise, we will not achieve our product, or we will not get our product. Enzymes are specific in their function due to the existence of active site. Very important point. We just enzyme ko be very specific. Ko mo time ti kono jago enzyme are very specific. We just very specific active site. Active site is very quite different. Ala enzyme substrate me chala la triangle. Be active site be la triangle. Me chala la rectangle active site bona. La active site ko design based on the substrate. Ko agent ko enzyme ko la go lo. Second allostric site. Allostric side now rather check side me now the check active side actually, but then take it out of the din dock on David. Allostric side met a gaka and then check a active side hello to gaka, but had a melody, geometric on there. Child is an additional binding side that does not have catalytic function actually. Gentle catalytic function, but take a binding side. Now the check active center, coactive center, you can take active side. Take a substrate binding site can meet you catalytic side. So then name function the regulation. So take a mutual regulator function on enzyme substrate binding site. So what it do is that allow me substance which a bind which active center from but a bang tell by not. But men belela take a conformational change betul ke active center kujume be active center berkulo lot kamu berkulo lot kami sabe sel mu don chere dalu kujeni won makana shebe kana lel keje ala jen ku learning regulation ku regulate garmana ku regulate active side and point the allosteric side. The term allosteric means the whole steering side. That means separated from catalytic and substrate binding side. Take good. Allosteric means change in shape. Again, let me go. So they change the shape of enzyme to facilitate regulatory function. Sometimes, trade a carcatin a different drug. Take a drug but some drug they may not have receptor finding. But in order for them to work, allosteric side will be called allosteric side. Substance management will bind allosteric side. No matter non-competitive inhibition. For example, aspirin, well, you're allowing to rejected. And non-competitive inhibition begin by mulel. The substance will bind the area of the active side, but by conformation of shape can make back go Walter Cocaine Bugany. Do you bind go tell me? So then turn a little allosteric side. Allosteric side. One activate in activator, allosteric activator, allosteric inhibitor. Allosteric activator, which a substance, which a allosteric, which a me substance bang, jogotong me, by activating active center. Me sub come allosteric, which allosteric inhibitor, me substance the bind there, but inhibit, which I don't inhibit, which I got, which I don't activate, I jacket. Which substance, me allosteric activator side, I load the substance more. By active center, by an activate cobarum with the cocaine. Me inhibitor, by active center, by an gag, by an inhibit cobarum, by go chabrum with the cocaine. So enzyme of region of the molecules, the allosteric side, that is not the active side or substance binding side, but is unique side where a small molecule binds and effects change in substrate binding side or activity occurring in the active side. So the binding of a specific molecule at allosteric cause change in mutual conformation of enzyme. This can cause the active site to become either more active when it is activator, mutual allosteric activator site. The enzyme is more active, or less active. Less active then means mutual allosteric inhibitor site by increasing or decreasing the affinity of the binding site for the substrate. The affinity binding site for the binding site active center. Which affinity decrease by inhibitor. Which affinity increase the activator. For enzyme, no matter fat enzyme again. Enzyme normally a protein. Part protein enzyme. We can make cofactor, can make coenzyme. We can go make enzyme. A pre-protein, we tell me that much enjoy. Coenzyme tank a lot of protein nature. Take a native, but be develop bucket to bar on market. No, it's a part but metabolic market. Enzyme, we tell cofactor inactive. So because it is inactive, you allow coenzyme. So a coenzyme is the protein part enzyme. Protein part enzyme without any cofactor ke prosthetic group that may be required for enzyme to function. So that enzyme is now what? Inactive. Chala coenzyme, la protein part. So being remember the protein part afo female day you so afo enzyme means the protein part of enzyme. So the specificity of enzyme is more dependent on apoenzyme and not on coenzyme. Very important. Again, quality enzyme a protein in nature, even though you can define kitchen to home check and money. I like specific enzyme kill. Again, apoenzyme quite is mine. And we go for example take at least two enzymes they may share same cofactor. But me work on one glycolysis with that banajak. I'll take a different enzyme, glycolysis. Check the first enzyme, which allow exokinase, glucokinase. Take cofactor, which allow magnesium. Allow magnesium mineral, Nikon. That one can service, can be metal. So magnesium gor exokinase cobacolet, metallic chabalet. Allow exokinase without magnesium are coenzyme. Meta magnesium colmatin, baculo enzyme, exactly the kilo. Okay. So, but no is will be as exokinase overlies by stimulate near step glycolysis. A chapel gyro stem over short glycolysis. Exokinase quit determine the protein party. Chia magnesium quit determine magnesium baby young yogalere. Non-protein country allow the non-protein organics, low molecular weight and isolizable substance I said with function can show coenzyme. Coenzyme they may be cofactor or prosthetic group. Cofactor are small organic or inorganic molecule that an coenzyme require for its activity. And a coenzyme go to door. Which a chocolate. Sorry. So apo enzyme, I mean cofactor, apo enzyme is a cofactor. Kala wa chalung low enzyme. Okay, so low enzyme. I think the yeah, very important here. So simple enzyme just consists of the peptide chain, like apo enzyme and simple enzyme. But Mr. Benla conjugated enzyme. But our enzyme can be simple or conjugate. When you say simple enzyme, tanajan amino acid Latin protein part only. Kula apo enzyme. However, metacomat cofactor ke prosthetic group by your low enzyme. So just remember low enzyme are 4 enzyme plus cofactor. A 4 enzyme then the protein part, which I call that. Cofactor can the non-protein part. The 3 additional non-protein component, for example, cofactor like iron, magnesium, magnesium, zinc. Really, we will use sometimes a different enzyme. But don't meet you queen of biochemistry, you will learn more enzyme to depend on cofactor. Most are mineral actually. So kakan cofactor, most cofactor, remember, most cofactor they are mineral. Coenzyme they are organic molecules, loosely non-covalent. That means they bind also with the enzyme non-covalently. So attach it to the enzyme and it's not consumed at the reaction and they are derived from vitamins. Can coenzyme. Prosthetic group are very 3 kakan non-protein, to contain a cofactor, coenzyme, and prosthetic group. They are very tightly or evenly covalent bound to the enzyme. Allow prosthetic group to can take a strict binding with the enzyme. But God are out here nearly. Binding then quay. And no cofactor mostly long mineral. Coenzyme no mostly like a vitamin. Vitamin not coenzyme are often regarded as second substrate. Yeah, we can call them as second substrate. They look like second substrate because substrate like a binding site for substrate binding site. Attack coenzyme, but take a guardabum there, never attack the coenzyme. So then call I shall second substrate on cause substrate, but which all know. Since they are painted with enzyme comparable with that of substrate. Coenzyme undergo alteration during the enzymatic reaction, which are later regenerated. Don't forget this one. Coenzyme can undergo alteration during reaction, but they will be regenerated later. But total enzyme during reaction, they will not be consumed after reaction. This is in contrast to separate, which is called by the product. Men la again. Ta go chikon men da gwan until go. Ta chikon good the measure ni kon with substrate gen da let check a second n coenzyme or cofactor or whatever it is men la ko garmulok. La chala co substrate ke da kon chala second substrate but no man kula da ja we na bar mi reaction to panyan coenzyme ba user but no ba onyo regenerate nya ko pas balo ngwade but no substrate betere ba nan ko product substrate balon wala substrate ba ko product jala da dia coenzyme do not decide enzyme specificity shalat content for enzyme mo ko design will be enzyme bala very specific. A particular coenzyme may participate in catalytic reaction along with different enzymes. You see, even a single coenzyme may be part of at least 3 enzymes. Okay. Don't let kill, for example, let's say glycolizers. Take hexokinase, one phosphofructokinase. Sometimes you may find, for example, magnesium can coenzyme, but that means it's not specific. Function will take a mutual regulation. For instance, NAT act as coenzyme for lactate dehydrogenase, can alcohol dehydrogenase, you see? Because here is specific, but these 2 enzymes are specific. In both enzymatic reaction, NAT is involved in hydrogen transfer. Tell you, so that means the coenzyme is not specific, but a coenzyme agent is specific of enzyme determined. Substrate, yeah, again the molecule acted upon by the enzyme to form product. A substance coenzyme the let go change from this one to further. For example, let's say if you have what we call sucrose carbohydrate, take enzyme which is sucrase. Enzyme sucrase during digestion, which you can make a sucrose, which is let's say it means the toir, means the digestion fruit. So there will be sucrose exactly. So before sucrose money. Balele ba use enzyme which is sucrase ba convert ba break down creo ba kono glucose kind of fructose. So which glucose no kind of fructose. What happened now? So you can take a further down to go maybe sucrose. So who was the substrate now? sucrose la substrate. So sucrose again substance ko enzyme la gide jen ko la jen substrate jen substance ko enzyme la ma goro bananga foroda. So sin model reaction are reversible, the product of the forward reaction becomes substrate by the reverse reaction. But this is very important. Most of the action enzymes, they are what? They are in equilibrium. When we say they are in equilibrium, this implies that when they are another topic, when they are in this issue, alan sucrose mi ko nan ba ko non glucose kan fructose. But mi kon fructose mat kan glucose si kon ko react back ba ngun kan ko non sucrose. So this is the end of the enzyme part 1. So I will continue with doing others for it. And thank you for your attention.